All right, well, welcome to the replacement for class on Friday. This is where we left off on Wednesday, talking about the muscle spindle and the stretch reflex. So we just finished up that, so let's talk about how that applies to adaptations to resistance training. So this is a classic study done back in the late 80s by Sale, and this was, just had a really interesting finding. And so pretty much what they did is they had subjects do a drop jump, right? So they stepped off of this box, landed, and then loaded, and then jumped back up, right? So that's kind of a classic plyometric activity where you have the loading phase, right? So this will be the loading phase, still loading, and then right between here and here is the transition, right? Transition from loading to the explosion phase when they're starting to push themselves up, right? And then here they actually jump and they get off the ground, right? So they did some drop jump testing with EMG on the gastroc, right? So they were testing electrical activity in the gastroc. And the nice part about this graph here is that all three of the picture and the two EMG tracings are time aligned. Okay, and so what we see here is we see pre-training. So this first line is untrained or pre-training. So this is pre, and this one is post. Okay, so they did resistance training between pre and post. Okay, and what they see here, so again, EMG in the gastroc. So before training, they landed, they jumped, and right at that point was when they saw maximal activation of the gastroc, right when they landed, right? So that was during the loading phase, right? And then they saw a little bit more activation here during loading, and then they did see a little bit of increased activity during the explosion phase when they jumped, okay? But not very much. Okay, and then then they did the training, and I think this was about 8 to 12 weeks of resistance training, doing some jump training, some other things, but also some true resistance training. And what they saw was that activation of the gastroc shifted more towards the end of the loading phase and into the transition phase. Okay, and so this delayed activation allows for us to get the force out of the muscle at the right time, right? We don't want the muscle to fire here where it did initially. We want it to wait. We want it to fire somewhere around somewhere in this range, so we can get all the force out of it when we're actually going to go do the jump up there. Okay, and that's what we see with training. So this has a lot to do with the muscle spindle. Okay, so let's think about it in this way with the muscle spindle. Okay, so in the throwing motion, if you were to cock your arm back to throw, okay, let's just think about it in terms of our pectoralis major muscle. Okay, there's lots of other muscles involved in throwing, but let's think about with the pecs to make it simple. Okay, when we cock our arm back in the cocking phase or loading phase, right, what we do is we stretch the pecs, right? Stretch or lengthen the pecs. Okay, so when we're doing that, we activate the spindle, right? The muscle spindle. So we stretch and activate that muscle spindle. And remember, when that muscle spindle activates, the action it takes is to contract that muscle that's being stretched, contract the pecs, right? And so when it contracts the pecs, and so really if you think about it, that muscle spindle is very important for transitioning from the loading phase to the explosion phase, right? And so if that muscle spindle fires at the wrong time, then you get a little bit of contraction, but it's not really what you want. You're not going to get maximal contraction out of that like you need, right? So if it's firing at the wrong time, like it did in this example, then you're not going to get as much force. If it's firing at the right time, so right when that pitcher wants to start bringing his arm forward, if that's when the muscle spindle fires, then we get a lot of extra help from those muscles, those muscle groups, or the motor units in the pecs. And so we have improved timing of spindle activation, which leads to two things, really. It leads to contraction of the agonist. That's a little messy. Contraction of the agonist at the right time. But it also leads to relaxation of antagonist at the right time, right? Because that's the other action of the muscle spindle, not just to contract the one that's being stretched, but to relax the one that's the antagonist of that. And so both of these things are helpful for getting more force, right? So those are both actions of improving that timing of the muscle spindle. In the Golgi tendon organ, right, this is going to kick in more during the explosion phase, right? So during the explosion phase, we're going to contract those pecs. Okay, and when we contract those pecs, we put tension, right? We create tension. And when we create that tension, if it meets the threshold of the Golgi tendon organ, it's going to cause two things, right? Relaxation of that pec, but then contraction of the antagonist of the pec. And so we don't want that until the ball is released, right? So we don't want that happening during the explosion phase. We want that after the ball is released from the pitcher's hand, right? So if we increase the threshold of GTO activation, then we're going to be able to withstand more force, more tension, really, before activating. Okay, so if we do that, then pretty much what we're doing is the two actions. Number one is allowing more force production, before inhibition of the pecs, or of the agonist, right? Okay, I'm running out of room here, so I'm gonna do it up here underneath the picture, and then number two. So come up there to number two. Sorry about that. Number two action is we allow more force and improved timing of the second action of the GTO, which is activation of the antagonist, right? So improved timing of antagonist contraction. All right, so we want the antagonist to contract, but we want to wait till after the ball is released, right? So that needs to be after release. Okay, so that's really what we're looking at when we apply these two adaptations, one with the Golgi tendon organ and one with the muscle spindle. Okay, so yeah, and this is kind of what we just walked through. So you can go ahead and fill this in on your own. But let me briefly do it here. So training adaptations for the muscle spindle include improved timing of activation, which leads to more force produced. Okay, more force produced by the agonist. Okay, so if we improve the timing, we get more force produced by the agonist. And we also want to, if we're doing that, at that same time, we also want to not contract the antagonist. Okay, so we got that. So both those things are going to give us more force, right? Both those things are more force. Okay. Secondly, with the Golgi tendon organ, increased threshold and also improved timing. Okay, if we get both of those things, then we get more force out of muscle before inhibition and less antagonist contraction. All right, that should do it. Okay, so a little bit more about that. The stretch shortening cycle, this is kind of just looking at that physiology, right? So this is the mechanism that allows for greater utilization of elastic proteins in muscle and the myostatic stretch reflex and full contraction of the muscle without inhibition by the GTO. Okay, so we get a actually reduction in the transition phase. That transition phase actually happens faster. So we move from loading to exploding faster. Okay, that's part of the adaptation. So again, that time between the eccentric phase and initiation of the concentric phase is decreased, and we get more force out of that. The muscle spindle responds faster with more power. That's improved timing. Okay, and the Golgi tendon and muscle responds with more force and is not inhibited by the GTO, and the GTO does not cause antagonist contraction too early. Okay, I think we're done with that one. We're going to move on to the structural adaptations next. All right, so here we go with the second part of resistance training, and this is the structural adaptations to resistance training. Okay, so we talked about this already. We talked about how those first 2 weeks are when we're going to see about half of our gains, and those are due to neurological adaptations. But then we talked about how it's about 4 to 6 weeks before we see any structural adaptations. And that's just the start of them. Okay. All right, so again, we've talked about this in the past, where the number one predictor of muscle cell or of muscle strength is the size of the cell, size of the cells and the size of the overall muscle, right? And so this just shows some skeletal muscle here before training. So we got kind of average sizes there. We can look at those, and then we look at after 6 months of dynamic strength training. We're going to see larger cells. Okay, so that's what we're talking about here. We're talking about muscle cells getting bigger. Okay, and this is obviously evident with the idea that weightlifting, in Olympic weightlifting and competitive weightlifting, is all about weight class, right? Because the larger the person, the bigger the more force they can produce, and the stronger their muscles are. And this is definitely why we have also a difference between men and women as well, because men are just typically larger. Okay, and you can see that the strength of a muscle goes up with the size, and this is really looking at the records for the snatch, and by weight class, obviously it goes up linearly with weight class. Okay, so if we're talking about how muscles are stronger when they're bigger, the question is, how do they get bigger, right? And so there are two possibilities for that. Number one is that cells get bigger. That's hypertrophy. Okay. Number two is that we get more cells. That would be hyperplasia. So we're going to talk about hypertrophy first, because I'll just tell you right now, that's what most of the research seems to point to, that the majority of the strength gains come from that increase in cell size. Okay, so there's 4 main factors that lead to hypertrophy. Number one is genetics, okay? But even with the perfect genetics for improving strength and increasing muscle size, exercise is still needed, okay? Second is the right mix of hormones, okay? But again, without training, even if you take anabolic steroids and you don't actually do resistance training, you get no hypertrophy. So again, that stimulus is still needed. Number 3, we need to have muscle damage and repair. Okay, so damage and repair is a key aspect of this. And so really, but again, we can't just bruise a muscle, right? We can't just give yourself a big quad bruise and then end up with hypertrophy, right? So that we need to have damage and repair that was brought on by the correct stimulus, and that's going to be, that correct stimulus is going to be eccentric contractions. Okay, and then lastly, which connects to that right there, is number 4, we need tension with a lengthening force. Okay, and this can stimulate protein synthesis. The most protein synthesis will be stimulated if we have eccentric contractions or that lengthening force. Okay, so hypertrophy occurs as a result of many of these processes, and these are some 4 key ones that we'll talk about. So let's talk about genetic influences first. So the main gene that dictates how well you respond to a resistance training program is called myostatin. Myostatin was discovered back in 1997 by McPherron, and McPherron pretty much was able to show that, first of all, myostatin is only expressed in skeletal muscle. Okay, it's a gene that's only expressed there. It's in the rest of your cells, but it's only expressed there. And they did this with mice, where they are able to genetically modify mice, right? And this is something you can do in research. You can actually order genetically modified animals, okay? And so in this case, they ordered myostatin knockout mice, where they actually just take the gene for myostatin out of the animals, and then grow animals this way. And you get those, and you can do experiments on them and see what happens to them if they don't have myostatin, right? And interestingly enough, these myostatin knockout mice weigh 2 to 3 times more than those of the regular ones. Okay, so key thing here, myostatin is a negative growth factor. Okay, the more of it you have, the less muscle mass. The less myostatin you have, the more muscle mass. Okay, so here's some interesting examples from those classic papers by Lee and McPherron. And here's a mouse with myostatin. Here's a mouse without myostatin. Okay, much larger. Here's what their legs look like, right? So there's the normal leg. There's the leg without myostatin, so just huge muscles. Same thing down here, right? And then interestingly enough, they tried this on a cow too. And look at these amazing hamstring muscles on the cow, right? So we can see some just incredible muscles. You can see hamstring muscles there, probably the 2 heads of biceps femoris there, and probably semi-membranosus there, I would guess, but I'm not sure, but just an absolutely ripped cow, right? Not what you would expect to see with a cow, but this is a cow without myostatin, so their muscles are able to hypertrophy as much as possible, and there's no limiting of that hypertrophy. Okay, so I'm sure some of you are thinking, like, why don't we knock out myostatin in humans too, right? And why can't we take a drug that inhibits myostatin? And that's definitely something that has been tried in animals. Problem is there are other negative side effects of that outweigh the positive benefits. So that was the genetic part. Now let's talk about hormonal influences. Okay, so a question to think about to start is true or false, do women release testosterone in order to increase muscle mass, i.e. elicit hypertrophy? Okay, and first of all, the question to start out with is, do women have testosterone? The answer to that one is yes. Women do have testosterone, but do they have a testosterone spike after a resistance training bout, and that leads to their muscle mass increase? And the answer to that one is no. Okay, so this one is false. We do not see that increase in testosterone after resistance training, but they still hypertrophy. Okay, so there's testosterone independent mechanisms in females. So bottom line is it's different. How men and women hypertrophy their muscle is different. Okay, so let's look at these different hormonal or endocrine influences that lead to hypertrophy. So again, we need the right mix of anabolic activity and catabolic activity, right? So the main hormones we need, number 1, is our catabolic one, right? That's cortisol. So we'll see a small cortisol spike post-exercise, and then that should go back down, and then we should see the anabolic hormones pick up. Okay, so we're going to see testosterone in males, growth hormone in males and females, and then possibly estrogen in females. And that one's not necessarily verified. There's some preliminary research that looks at that, but we're not quite sure about that yet. But definitely testosterone in males, and this in both, and growth hormone in both males and females. So the amount of these hormones that we get to increase is highly dependent on the type of exercise that we do. So the large muscle mass, multi-joint resistance exercises will cause larger changes in the hormonal status. Okay, so the more muscle mass involved, then larger swings in these hormones, and then larger gains in hypertrophy. And this is exactly why Olympic weightlifting is so popular in sports. It's because you're using so much muscle mass, and the more muscle mass involved, the more swings you're going to get in these hormones, and the more hypertrophy you're going to get. So Olympic weightlifting is Olympic weightlifting is key for causing these large hormonal swings. Okay, so number one, cortisol. We've talked about this one before. It causes breakdown of protein, proteolysis, right? It also causes breakdown of fat. Okay, both of these together are going to lead to more energy availability in the blood. Okay, but that's not all it's doing. That's the part of the stress response, right? So stress causes this. Exercise is one of those stressors that causes that. Okay, so that proteolysis, that's the catabolic part. Okay. Secondly, testosterone is obviously very important, and there's two separate mechanisms that testosterone uses to lead to hypertrophy. Okay. Okay. Sorry about that. I had a brief interruption. Christian was starting to yell in the background. So let me see where I was. Okay. So again, testosterone has two separate mechanisms. Okay, for increasing. For being a part of hypertrophy. So number one is the fact that it's a direct transcription factor. And you guys probably learned about transcription factors either in Bio 210 or a little bit in Bio 130. A transcription factor is something that goes into a cell, gets all the way into the nucleus of the cell, binds to the DNA, and starts the process of transcription, which that's to make RNA, right, which is the first step needed to make a protein. So we've got to make RNA before that RNA can come out to the rough ER and to make a protein. Okay, so testosterone directly causes the initiation of protein synthesis, right? So that's making more proteins is making more stuff inside a muscle like actin and myosin, right? So that's the first thing. It's a transcription factor. Okay. Secondly, it activates these things called satellite cells, and that's shown in this one here. Testosterone activates satellite cells. Satellite cells are cells that join up with muscle cells and act in two different ways. The first way could be to create a new cell that would lead to hyperplasia, or secondly, that satellite cell could join in with an existing muscle cell and cause hypertrophy. Okay, so here's just some classic studies looking at testosterone levels in both males and females and how they relate to strength. So on the x-axis here we see age of the participants in years. So this is looking at kids through adolescence, 8 to 18, or 19 on the top one. The top chart looks at their strength. males and females, and then the bottom part looks at their testosterone, right? So right at about 11, we see a testosterone spike. Between 11 and 13, we see the testosterone spike in males only, not in females, right? And right about that same time is when we see the increase in strength, right? So in males, we also see an increase in strength in females. It's not quite as drastic at that same point, but it definitely increases as well. So this is just showing that testosterone levels and strength are highly correlated in males. Okay, secondly, this is looking at the effects of 8 resistance exercises, 10 rep max, 1 minute between rest. Okay, so what we see is that testosterone levels in males and females here. Okay, so we see pre-exercise testosterone levels, about 18 to start. All right, during exercise, it's gone up almost double, but not quite, but to about 31. Okay, and then post-exercise, we see that testosterone level stays pretty high, and even after an hour, it's still elevated to about 21 versus the 18 initially. So just showing that one bout of exercise, resistance exercise in males, leads to a testosterone spike. In females, it does not. Okay, so which hormone is the most common one taken to actually, when we think about performance-enhancing drugs in baseball athletes, and mainly baseball athletes is what it's been lately, to increase muscle mass. Okay, and yeah, I'll give you the answer right here. So it's the precursors to testosterone. If they could just take pure testosterone, that would be better, but that's too easily detected. They've done that in the past. Growth hormone has been played around with, but the thing that's been happening lately is these precursors to testosterone. Okay, so hormone precursors, also known as prohormones, also known as androgens. Okay, these are the same types of chemicals that are released by the adrenal gland in males and females, and they can be turned into testosterone or estrogen. Okay, so one of the most common ones is DHEA. It's also known as androstenedione. Okay, dehydroepiandrostenedione. Sorry, so that's that one. It's going to convert into androstenedione, which is eventually converted into testosterone. Okay, so these can definitely have anabolic effects. They're not as potent as pure testosterone, but they can do it, and they're not as easily detected by drug screening. Okay, so next, growth hormone is very important, but growth hormone alone doesn't stimulate hypertrophy. It actually triggers other hormones like insulin-like growth factor to cause the actual protein synthesis. Okay, so growth hormone is released from the pituitary, right? And then it goes to the liver, cause the liver to release that IGF-1. That's what's going to cause growth in tissue. Okay, so there's kind of 2 steps to it. So the 2 stimuli that are important for its release here, number 1 are exercise. We exercise more, we get more growth hormone release. Number 2 is sleep. So sleep, and getting the right amount of sleep, is incredibly important for getting the right as much hypertrophy as you want. Okay, and then putting all of this together a little bit, different mechanisms involved with protein synthesis in muscle. One of the main things that's been discovered recently is this molecular mediator called mTOR, and there's what it stands for: mechanistic target of rapamycin. Okay, so it's seen here. The key that we've learned, physiologists have learned in this kind of research, is that we have to activate mTOR to get skeletal muscle protein synthesis. Okay, and there's 3 separate ways to do this. Okay, number 1 is insulin, number 2 is resistance exercise, and 3 is amino acids. Okay, so we can see that right here. So insulin comes and binds an insulin receptor. Okay, that activates another molecular mediator called AKT. That directly goes and activates mTOR. Okay, that's number 1. So how do we get insulin release? We take in glucose, right? So most of your protein shakes have glucose in them, and one of the main reasons for that is because It tastes better when there's sugar in it, but there's also some evidence to show that maybe mixing some sugar in with protein can have an additive effect and leading to more activation of mTOR because of this insulin pathway. Secondly, resistance exercise itself leads to a buildup of phosphatidic acid, and that directly activates mTOR. And then lastly, we see leucine right here, right? And leucine is such a popular thing in whey protein, right? And so leucine is a direct activator of mTOR as well through this pathway, right? So we see that. It's also easily digestible. Whey is easily digestible and has a high percentage of leucine. And so if we do all three of these things, have that insulin spike, resistance exercise, and take in the right amino acids, then these things are going to lead to the most mTOR activation, lead to the most skeletal muscle protein synthesis. Very important figure from your book. Make sure you know that one. Okay, and we're going to stop it there. We'll pick up the rest of this on Monday in class. Thanks for listening, guys, and we'll see you on Monday.